In this video, we will discuss about how this high fatty acid diet could lead to a pre-metastatic stage of the pancreatic cancer development. So in this video, we will discuss the one of the characteristics for this pre-metastatic stage is this overactivations of this platelet uh, because of the high fatty acid diet. So let's start with this panel A, where we checked two different conditions. First one is treated with the high fatty acid diet, another one is the control, and in this case we react the we react these platelets with the thrombosis for 5 minutes or 15 minutes, and we are able to see that high fatty acid diets are more prone to activations compared to the control, and these things are quantified in the panel B, where we can see that the activations of these platelets is significantly higher when they are either treated with for the 5 minute or for the 15 minute compared to the compared to the conditions where the control cells are treated with thrombosis for 5 minutes or 15 minutes. And another thing that they check that could cause the pre-metastatic stage is this more aggregations formations, which is somewhat related to the ATP release, and they are able to see that in the high fatty acid platelets has more ATP release compared to the control case. And then they also check the platelet adhesions through this fibronectins FG, and they are able to see that these platelet adhesions are also higher in the case where HFD diet is used compared to the control case, and these things are shown in the figures in this panel E, where we can see that as the treatment with the thrombosis increases, they see a more adhesions of these platelets on the fibronectins compared to the control case. And when they check the different stages of this platelet activations, they are able to see that in the case where HFD treatment is done, they are able to see more of this fibronectins compared to its control case. That can be seen here. FP is phalopodian and SP is spread. So we can see that spread is increased, and also this phalopodium is increased in the case of this HFD diet compared to the control case. And these things are quantified later on in the panel G, where we can see that the FP levels are significantly higher in the case where HFD diet is used, and also these SP levels are also higher. And these two are the main characteristics, one of the main characteristics towards this platelet activations. And then they check the mechanisms by which the platelet activations might have occurred, and they are able to see that the phosphorylation HFD treatment compared to the control case, and similar results they have seen for these PKC phosphorylations also. Here we can see that PKC phosphorylations there is not much significant difference that we can see, but for the SRC phosphorylations we can see the significant difference, and for this tail in phosphorylations we can see the significant difference when the cells are treated, when the platelets are checked, which are treated with HFD or control. But for the case of this FAK and AKTP, in AKTP also we can see is a increase in the signals. AKT phosphorylations also we can see an increase in the signals, but for this PKC phosphorylations and for this tailin phosphorylations, even for the PKC, even for this FAK phosphorylations, we can see a slight increase in the signals. So all this overall this panel I, J, K, L, M indicates that the mechanisms that includes these SRC phosphorylations, AKT phosphorylations, FAKP phosphorylations, and tailin phosphorylations plays a role towards the overactivations or activations of the platelets, and that might interact, and we know that the tumors usually interacts with the tumor microenvironment, and these activated platelets might enhance those interactions and can lead to the pre-metastatic stage, which are usually observed in the breast cancers. So I hope you understood how they confirm this high fatty acid diet could enhance this platelet activations, and we will discuss how this will lead to the pre-metastatic stage in our subsequent videos. So that's all for this video. If you need to understand more about their experimental results in detail, you can go through the reference that has been provided in the description. Thank you for your time.